Okay, welcome back from break. I've heard some of you say you had a stressful break. Like, that's not the way it was intended to be. Hopefully you had a chance to do something fun the last 4 days. We do have an exam on Friday. So I did previously forget to post some practice questions. I did that last night. So there's the tab on Blackboard that says exam objectives and practice questions. There's about 12 questions there for you to practice on. I also did just this morning put off the chapter 15 homework until the next exam. I think a few of you did it already, but there you'll be ahead the next time. So what we're going to talk about today, none of this will be included on Friday's exam. So remember, Friday's exam starts with, there was just a few PowerPoints on horizontal gene transfer. So that's where it begins. And then there'll be one question on horizontal gene transfer, and then into the viruses, the microbial control, antibiotics, and chapter 14, mechanisms of pathogenesis. So yeah, any questions that you have that maybe I could answer now before we keep rolling? Anything in particular that maybe anything that you wanted to bring out? Okay. I do have office hours tomorrow. I will be in my office before the exam all morning on Friday. So if there's anything that you want to go over again, don't be afraid to stop in and see me. Okay. Let's see. Oh, and also remember, drop date is Friday. So I think I made an announcement to my lab people, but remember, if you're thinking about dropping lecture and you want to stay in lab, you got to talk to your lab professor about that. And just remember, after Friday, you need some kind of emergency to come up for me to okay you dropping it. So beyond, I'm not doing very well. So that we need to think about that this week. Okay, so again, see me with questions about that. All right, so as we move forward, I think this is some of the more interesting material that we talk about this semester, just the process of infection and some kind of, we would say, the language of epidemiologists, things that people have been thinking a lot about the last couple of years as we've come through a pandemic. So this is a fairly short section. We'll get through this and we'll start on innate immunity today as well. Okay, so what is epidemiology? Epidemiologists are going to study when and where disease occurs, and not just infectious disease. The Centers for Disease Control in Atlanta, the CDC, they also think about things like gun violence and suicides, other aspects of public health and not just infectious disease. So epidemiologists are doing a lot of field work. They're asking a lot of questions to try to understand how diseases are passed, what causes them, etiology. It's a lot of statistical analysis that's involved there. Some terms that we use: incidence and prevalence. These are really two terms that characterize morbidity. Remember, we talked about the difference between morbidity and mortality. Mortality is the people that are killed by a disease. Morbidity is just the people that are ill from a disease. well, we could kind of further zero in on incidence and prevalence. Incidence is the number of new cases, people that are newly contracting a disease, and we might describe incidence, per week or per year, for instance. And then prevalence is the total number of people that are alive at any one time that also are living with the disease. So as an example there, in your book, they talk about incidence and prevalence of HIV. So this bottom line is the new HIV infections. So that's going to be incidence. And the red line, this is the number of people that have HIV infections. That's the prevalence. So you can see it's kind of interesting. The incidence peaked here in the mid 80s. We had a peak in the prevalence actually a little bit later than that. It used to be that people didn't live very long once they contracted HIV. But now people are actually living close to a normal lifespan when they have HIV. So we've gotten really good at treating that. There's no cure. It's one of the infections that hopefully we'll have time to talk about at the end of the semester. But that's why the prevalence has really skyrocketed, because people aren't dying right away. They're living with it. Okay. Some other terms that epidemiologists like to use to describe infectious disease. If they're sporadic, that just means that you get a few cases here and there. They're not geographically clustered. An infection like anthrax, for instance, that's not passed from person to person might be just sporadic. You get a case here or there. An infection that's endemic is going to be constantly with us, usually at a low level. We'll pick that term apart just a little bit more. You can have seasonal fluctuations, but something like influenza, we're always going to have that. We're always passing it around. We're never going to totally get rid of it. An infection that's epidemic, an unusually large number of cases in a short period of time. And then a pandemic is when you have an epidemic on more than one continent. All right, so I'll come back to that link in a second, but this is just kind of graphically, maybe will help explain those. But again, sporadic, a few cases here or there. Endemic, colds are endemic, basically. Here's an epidemic, lots and a much higher number of cases, usually clustered together in one spot. The question is, how do we know what is a unusual amount of cases? How do we know if it's higher than normal? Oh man, let's see. I want that bigger. Okay, what button do I need to press here? Any ideas about that? It's at the top. Oh, thank you. All right, good. There we have it. Okay, so this is a link to they used to call this the Morbidity and Mortality Weekly Report. They changed the name, but it's nationally notifiable diseases. So in this country, we have a list of diseases that if you go to the doctor and you are diagnosed with that infection, the doctor by law has to report that case to the CDC. Okay, so we have a number of infections that we keep track of, so that we know what's going on. So we'll notice if we get an unusual high number of cases in any one spot. So some of these you'll recognize. Let's look at measles and malaria here, because we talked about malaria a little bit. So this is for the week ending October 15th. So this is recently here. You can look at, so you can look at the current week. So right at the top there, I think my pen's not working because I'm on internet here. but you can see in the current week for malaria in this country, in the US, we've only had 3 cases. Okay, and then that previous 52 weeks maximum in the past year, what's the week where we had the most number of cases? So there was a week that we had 56 cases in the US. Year to date, this year we've had 1,084. Compare that to last year at this time, 1,206. Okay, so pretty close to about the same number. And then you can look up per state how many malaria cases. Next to that, the measles. So one that we do have to keep a pretty close eye on to see, what's going on. If we have a large number of cases, we need to, make sure people are getting vaccinated. Again, pretty low level of measles right now, and we keep track of that. Cases that we think were brought in, people traveling from other countries, and people that maybe got it right here. So this is how we know if, again, how do you know if cases are higher than normal? Well, we keep track of that stuff. And just to pick apart that term endemic a little bit more. So this is from your textbook, OpenStax. Like I mentioned, it can be seasonal fluctuation. So there's a seasonal baseline there. Cases are going to peak in the winter, early spring months. That's what we see every year. And then we have kind of a level here, an epidemic threshold. So if cases get above a little bit higher than that, then we would correctly call it an epidemic. The last year that I remember that they called flu epidemic was 2018. So we had a big spike that year. I remember that pretty well because we had a lot of absences that winter. About every 10 years, we have a flu epidemic, whether that's just because, immunity kind of wears off a little bit, I don't know. But we are due for another peak like that. Since 2018, we have been. In the last couple of years, everybody was masked. We didn't see much flu. It could be a bad year. So just saying, if you haven't had your flu shot yet, still time to get it. It's been in the news the last week or so, young kids and babies in the hospital with respiratory viruses. So COVID, influenza, and that's RSV. So we've got vaccines against COVID and influenza. So they're really encouraging parents with young children to get at least those vaccines. And we don't have a vaccine to RSV, but filling up the hospital beds with those infections that are again normally considered endemic. Okay, at times, if they're more than kind of the normal level, we would consider them an epidemic. So it kind of came up. All right, when are we going to consider COVID an endemic disease and not consider it epidemic? Well, according to CDC, when an infection is circulating at kind of a predictable level, then we would consider it endemic. But it depends on factors that are not just straight numbers. So if you think it also depends on what level of disease, meaning kind of numbers of infections, officials and individuals are willing to tolerate. So when I say willing to tolerate, that means like, are people willing to change their behavior because of that infection? For instance, are they willing to wear masks? Are they willing to not congregate in big places? And it also depends on how the virus evolves. Okay, if we are speaking of COVID, for instance, if we were constantly coming up with or constantly getting exposed to new variations that people were getting COVID over and over and over again, which we were worried that, we've seen that happen. It seems the evolution has slowed down a little bit. Then we would still consider an epidemic. But a big hallmark of when we decided to call COVID endemic just is by the lack of social disruption. Okay, so for the most part, people are not changing their behavior anymore because of COVID. And of course, that varies by individual, but for that reason, more likely, most people would consider it endemic now rather than an epidemic. All right, so when we finished last time, we were talking about portals of exit. What happens to a pathogen when it leaves a host's portal of exits? Sometimes it might immediately get transferred to a new host. But oftentimes pathogens might have somewhere in the environment or maybe a different host that they kind of hide out in a while. So we call these places reservoirs, all right? Sites where pathogens are maintained. And a reservoir can be living or it could be non-living. Typically, if it's living, then we would say the individual may or may not have symptoms. Okay. So they may or not, they actually may or may not be affected at all by the disease. We see both ways. So when you have an animal that's considered a reservoir, then the disease is going to be considered zoonotic. Okay, so we talked about that term before. Zoonoses is a infection that's has some kind of animal reservoir, and lots of infections that we contract because we're in close association with animals. So usually, not usually, let's say all the time. Like, let me make this a little bit darker. Pass to humans when we are in close association with an animal. So something like a bite, for instance, bite from a rabid dog, we can get rabies. Bats are a reservoir for many, many different kinds of infections. Humans don't very often get bit by bats, but we can maybe come in contact with bat guano, bat species, and get in certain infections like rabies that way. Ebola, we think, has a reservoir in bats, probably COVID as well. So bats is just one example of an animal reservoir. Let's see. Humans can also be considered reservoirs as well. And when humans are not displaying symptoms, then we call humans carriers. Okay, so when humans are causing symptoms, then we would more just consider them a way of a mechanism of transmission. But if a human's not causing any symptoms, if they are a passive carrier, that just means the individual is not infected. So an example there would be maybe a healthcare worker that doesn't wash their hands between patients and inadvertently transmit some pathogen from one patient over to the next room. Okay, that's just a passive carrier. A human can be an active carrier that is going to be infected. This is an infected individual that can pass the disease to others. So remember, this is an individual that's not showing any symptoms. So it could be that that individual is just in that beginning stage of infection where when they're not showing any symptoms yet. Incubation period, that wasn't coming to me. So of course, COVID people could be not showing symptoms and still be infected, not just showing symptoms yet. Another classic example is typhoid Mary. So people that have typhoid fever caused by the bacteria Salmonella typhi can contract the infection, and then a very small percentage of them keep an active infection in their gallbladder. And those bacterium are still, even though they're not making the individual sick anymore, they're still shedding that bacterium in their feces. So back early 1900s, before we routinely treated our water supply, this was a huge problem. This woman, Mary Mallon, was a active carrier. She wasn't sick herself, but she was shedding the bacterium in her gallbladder. Probably didn't wash her hands after she went to the restroom. And she worked as a cook for families. And she would, every family that she went to, they were having outbreaks of typhoid fever. People died. That infection has about a 15% mortality rate. Finally, they traced all these outbreaks to her, and she was really belligerent about it and wouldn't believe that it was her that was causing the problem. And she ended up under house arrest for the last like 20 years of her life. So classic example of a human active carrier of an infection. Reservoirs can also be non-living. So we could have pathogens persisting in the soil, more likely pathogens that can make endospores. So things like botulism or anthrax or tetanus can persist in the soil for long periods of time. A lot of different pathogens can be maintained in the water. Either the pathogen will grow there, or it can just be found there because of some contamination of human or animal feces. Lots of different examples. We've talked about Cryptosporidium. It's a protozoa that's maintained in water. Typhoid fever, like I just mentioned, Giardia, lots of different things that can be maintained in the water. Cholera, that's another example. And then food can be considered a reservoir, especially if the pathogen is maintained there for a long period of time. Any foodborne illness, things like raw milk is a really good common example before we pasteurize our milk supply. So this picture, I didn't explain this. This was another typhoid fever example. There was a town back in the, I think this was from our old textbook, this little inset, in the 1930s, a town that was having a spike in typhoid fever. And they finally traced that back to a worker. They were laying new pipes for the town's water supply, and they found out that a worker was an active carrier of typhoid, and he was habitually peeing into those new water pipes that he was helping to lay. So it's not many infections are passed through the urine, but that's one of them that can be. So, okay. So if we think about how pathogens are passed from person to person, kind of the first kind of transmission that we will think of is contact transmission. So one person comes in direct contact with another person. That could be through kissing or intercourse, or just direct contact like a handshake. Droplet transmission is also considered direct transmission. So those large drops of saliva that are carrying viruses and bacteria that are coughed out or sneezed out even sometimes just talking to somebody, you spray a little saliva. Okay, closer than 6 feet. Sorry, closer than 6 feet. All right, and then indirect contact transmission involves some kind of non-living object. Remember, we call that a fomite. Okay, so it could be a doorknob, it could be a computer keyboard, it could be bed sheets. Yesterday, I spent a few hours preparing some information for you all on monkeypox. So hopefully that's one that we'll get to at the end of the semester, but bed sheets can transmit monkeypox. Okay. So that's a fomite, non-living object. And of course, COVID can be transmitted all of those ways. So usually an infection doesn't have just one way, it might have a more common way. So for COVID, most commonly droplet transmission, when you're closer than 6 feet away from the person that is ill. Sometimes transmission is by some kind of vehicle. Okay, so water, food can both be considered vehicles. We considered those reservoirs too. So here you have to kind of understand how the life cycle of the pathogen, and if maybe they're only briefly in that water or food, then we would consider water the vehicle. If they spend more time dividing and living for longer, we might consider those things reservoirs. But again, water is really common, especially for infections that are fecal oral. So we drink some contaminated water. It's been contaminated by some feces. It's only in the water kind of a short time, probably. Foodborne here, usually we would consider food a vehicle if it was the worker that was preparing the food, maybe that had a staphylococcus infection on their hand, and some of the food got on that and contaminated the food just for kind of a brief amount of time. That's enough to get somebody ill with food poisoning. Anything that's airborne, we could say by dust in fine particles, or what we would refer to as droplet nuclei. Or you could call that aerosols, basically. So those are the really fine particles. Here's an unstifled sneeze. When anybody sneezes out into air like that, really teeny tiny particles might stay suspended from that sneeze for quite a long period of time. Some pathogens can ride on those really fine particles, and then somebody can be ill if they're greater than 6 feet away from the person, say, that sneezed. If they become aerosolized, again, they hang in the air for a long period of time. So maybe not as many infections are considered truly airborne. Tuberculosis is, hantavirus is a virus that is found in the feces of mice, feces and urine of mice, and that gets aerosolized in the dust. And people can inhale hantavirus. Measles, airborne. COVID can be airborne, but we don't think it's commonly transmitted that way. Okay, so again, if you're exposed to somebody and you're greater than 6 feet away and you still get it, that's by considered airborne transmission. And lastly, vector transmission. Vectors are usually, we call them insects. Vectors are usually insects, although your book says that occasionally animals can be vectors. Typically, we would consider animals reservoirs and not vectors. So I don't really like that definition, but they're usually arthropods, usually insects that are carrying the disease-causing microbe. If it's, and then we consider that vector either a biological vector or a mechanical vector. A biological vector, that just means that the pathogen is reproducing growing in numbers within the insect. So you'd have to know something about the life cycle of the pathogen, or to kind of understand whether the vector is a mechanical or biological. But lots of examples of biological vectors: mosquitoes, when they carry malaria, because we know that part of that life cycle of that protozoa has to develop within the mosquito. Bubonic plague that's a bacterium, Yersinia pestis, that's carried in the fleas that live on rats. Part of the life cycle of that, again, that bacteria will grow up and multiply in the fleas. So that's, again, another example of a biological vector. A mechanical vector, it makes it sound like it's a robot or something, but again, this is an insect that just passively carries the pathogen. Okay, so the best example I can think of that is maybe a fly that lands on a pile of manure in the farmyard and then flies off to the picnic and lands on the potato salad. Okay, so it could transfer some E. coli or whatever in the manure to now maybe cause some foodborne infection. So that bacterium was just being carried. It wasn't really growing in numbers or anything. Right. So that's pretty much it for chapter 15. There, I just included this table that has some just some more examples of some zoonoses. Just getting you used to using that term reservoir and different modes of most common modes of transmission. So mostly just kind of some terminology to understand kind of the language of how we talk about how diseases are passed. Okay, any questions there? All right, so we will move on. We are going to be talking about immunology for the next couple of weeks. Okay, so this is a big chapter that gets sometimes a little bit more conceptually difficult, especially when we get to the adaptive immunity. But what does the immune system do? Well, we are being bombarded constantly by many different pathogens, viruses, bacteria, fungi. Don't memorize those numbers. Obviously, they're probably a little bit off. I copied that a long time ago. But our immune system is constantly working so that we don't even know that we're exposed to these things most of the time. The immune system also helps repair wounds. There's some inflammation, especially that has to take place before the repair process can begin. Our immune cells are constantly cleaning up, blood cells that are dying, other kinds of cells that die and have to be replaced. Unfortunately, or I should say, fortunately, there's a lot of checks so that the immune system doesn't target our own cells, but that sometimes things go wrong with that. And then we can have autoimmune disease. And we don't really talk much about that in this class. We'll talk more about that if you take immunology, a class that's offered here at Grand Valley. I didn't post this because I didn't want you to freak out that, oh, I have to memorize that. The point that I want to make here as we get started is that immunology is pretty complicated because a lot of these cells that are important in the immune system, like B cells and T cells and macrophages, they have lots of different functions. Oftentimes those functions overlap. And the way they signal each other, we call those signals cytokine signals. There's many different cytokines, and those all overlap in function. So it's good for our health that we have cells that kind of do the same thing in case one kind of cell isn't working. It makes it hard to learn about. If I say, let's talk about macrophages, well, it's hard because you got to understand how other cells are acting to understand what macrophages do. So it is complicated. We'll do the best we can. Just remember that everything is more complicated than what I'm going to talk about. So we're going to kind of talk in generalities And there's a lot we don't know about the immune system as well. We're really rapidly learning about it, but just kind of bear those things in mind. Okay, so a little overview of what we're going to talk about first are our innate immune defenses. We call them innate because that means these are defenses that we kind of naturally have. They don't change the second, the third, the fourth time we're exposed to that pathogen. We pretty much have the same response every time in general. So what does that encompass? So we're going to talk about some physical barriers. First one that you think of is skin. Okay, that's a great physical barrier that we have against infection. We'll talk about a few other things. Chemical defenses. There's a lot of chemicals that I'm not going to get into that even your textbook talked a little bit more about. We're going to spend some time thinking about complement proteins and how they act in our blood. We'll talk about different cytokines a little bit, the role of inflammation. Those are all part of our innate immunity, as well as how different cells work. Granulocytes like neutrophils, agranulocytes, B cells, T cells. B cells and T cells, actually, that's a poor example because we typically consider what they do as part of our adaptive immunity because they mount a very specific response to very specific invaders. Okay, so that's just a little overview of where we're going. I'll ask you a little thought question here before we get started. Okay, so knowing what you do about innate and adaptive immunity, and I'm assuming that you don't probably know a whole lot, if you had to make a very hard choice, which would you rather do without? If you had to do without one, would you keep your adaptive immunity and get rid of your innate, or keep your innate and get rid of your adaptive? Okay, what do you think? Oh, question. Oh, assume that you still have your skin intact. Yes, let's not do without that. Okay, good question. All right, how many of you think I will get rid of my innate immunity? Show hands. Okay, a few of you. Okay, good. How many would get rid of their adaptive immunity? Okay, a few more of you raise your hands. When I have asked this by clicker question, I typically get like 50-50 response. Okay, that's good. It turns out that there is a correct answer there. So let's explore what that is. There was a very interesting experiment done, I don't know, years ago using knockout mice. And knockout mice it's really a cool thing that we can do. We can breed mice to have some kind of knockout. So they are not able to do something like, oh, they can't make this kind of cells, or we've knocked out some gene. So they've designed mice, is a better word, that are deficient in certain aspects of our immunity. Okay, so if you inoculated some of these mice with some pathogen, normally in terms of the level of microorganism that would rise up, and then mouse with normal immunity, they would fight it off, okay, and return to health. But if you took some knockout mice here, these are called SCID, or severe, severe combined immunodeficiency, I believe is what that stands for. But these are mice that have PMNs, or neutrophils, and macrophages. So those are two really important cells in our innate immunity. So they have those cells, but they don't make T cells and B cells. Those are the important cells in our adaptive immunity. So they can kind of curb the level of microorganism, but they can't quite all the way fight it off. But at least they can kind of suppress them from reproducing uncontrollably. If you take mice that do not have those neutrophils and macrophages, but do have T cells and B cells, basically they're not able to suppress that growth at all. because as it turns out, in order for the B and T cells to get activated, they need help from those macrophages, especially in order to kind of present that particular pathogen to activate them. So if you said you'd rather do without your adaptive immunity, you would probably live a little bit longer. You're still not going to be in great shape, but you would probably live a little bit longer. So good. All right, so let's take a look with the time that we have left, thinking about some of those physical barriers. First thing that we come to is our skin. And we mentioned our skin a couple lectures ago when we were talking about portals of entry. And I basically said, this is a really bad portal of entry for pathogens. Our skin is a pretty good barrier. So some of the reasons why. We mentioned this before, but the top few layers of our skin are made up of dead cells. So we talked about how pathogens have to be able to adhere to their target cells. And if those cells are dead, then the cells aren't going to be presenting the kinds of receptors that the pathogens need to really hook onto them. So they have a hard time adhering. Even if they do adhere to the dead cells, then they're out of luck because those cells are continually being sloughed off. So we call that desquamation. I think there's two M's there. I think I spelled that wrong. Okay. So desquamation, we're constantly losing those outer layers of our skin cells because they're dead. Okay. So that makes up, you know, most of the dust that ends up in our house, our dead skin cells. Okay. Our outer layer of skin, that epidermis, also has a lot of keratin in it. That's a structural protein. Okay, it's pretty tough. Okay, that outer layer is pretty tough. It's difficult, again, for pathogens just to pierce through it. We might as well talk about some chemical defenses, even though that's not quite how the book presents it. Deeper in our skin here, we have these oil glands, also called sebaceous glands. And they are going to produce sebum. Which is kind of an oily substance. It's going to lower the pH of the skin. Also makes the skin pliable. All right, so lower pH makes it kind of an inhospitable place. Sweat glands are going to secrete sweat, which is very salty. Okay, so a salty place that's going to dehydrate pathogens, bacterial cells. The pH is low. We also have cells that are patrolling around that we haven't talked about yet, but we'll mention them here. Dendritic cells. Okay, they're patrolling around, phagocytizing invaders. Okay, and there's a lot of chemical defenses as well that I probably won't talk about very specifically, but the skin in general is a pretty inhospitable place, not a very common portal of entry. So here's just your book's figure. These sebaceous glands, they're usually associated with hair follicles because that is one place that sometimes pathogens can gain a foothold around the hair follicle. But again, that oily sebum is going to help prevent that. Our mucous membranes, we said that mucous membranes are a really common portal of entry. We do have defenses here, they're just not quite as good. So what kind of defenses do we have? Remember, mucous membranes, these are going to line all of our openings to the outside, like our respiratory tract, urinary tract, digestive, reproductive tracts. And just an FYI, when here, when we use mucus as an adjective to describe membranes, we spell it M-U-C-O-U-S. When you're talking about mucus as a noun, then we spell it M-U-C-U-S, which is really annoying that we spell a word differently. But just so you know, I didn't misspell that. The epithelial layer on mucous membranes is pretty thin. So we only have sometimes a single layer of cells there on the outside. And those cells are alive. But even though they're alive, they are continually being sloughed off and replaced, just not as quickly as the outer layer of our skin. There are cells that secrete mucus in those mucous membranes, and those are cells called goblet cells. So if you look at, this was the best figure I could find of this. They didn't put one in your book, but here's a goblet cell here, and you can see the mucus that's being secreted. We produce about 1 liter of mucus a day, which seems like an astonishing amount. Not saliva, that's different. Just mucus. A lot of it in our respiratory tract, at least when it's produced, actually ends up getting kind of moved out and then swallowed. But that's an astonishing amount. In the respiratory tract, we have another kind of cell along with the goblet cells here called ciliated columnar cells. Okay, so here in this picture, you see the cilia. Here's the ciliated columnar cells. Again, these have cilia. And remember, the cilia are they've got the same structure as a eukaryotic flagella. They move in a wave-like motion. And their job is to move that, all those pathogens and dust and whatever that get caught in the mucus, and to move them up out of the respiratory tract. So we call that movement the mucociliary escalator. Okay, so again, that's when cilia is moving pathogens out back into the mouth and end up swallowing most of it. Okay, so if you are a smoker, then those cilia don't move as efficiently, or sometimes that smoke causes changes in those ciliated columnar cells so that cilia are not produced. Okay, so people that smoke don't have as good kind of physical and chemical response here to respiratory pathogens, and more likely to be ill with respiratory pathogens. There's also some antimicrobial peptides. I won't get real specific there. One that we have talked about is lysozyme. Remember, we talked about how some bacteriophage will carry with them lysozyme. That's a protein that's able to break those bonds between peptidoglycan sugars. So it helps them enter bacterial cells as well as leave it again when it's time to lyse the cell. We have lysozyme in our mucus, in our tears, lots of places, lots of our bodily secretions have that. It's just a mechanism against gram-positive pathogens. Okay. So again, just some visuals. Here's a visual of a goblet cell. Lots of secretory vesicles there. Lots of Golgi packaging that mucus that's made in those cells. And here's a close-up. Looks like a scanning electron micrograph of some cilia in our respiratory tract. Okay. To end with today, also there is a big role for our normal microbiota. So we started to discuss this a couple lectures ago, but we know our normal microbiota helps us outcompete pathogens. So we call that microbial antagonism. So that just means that our resident microbes help to outcompete pathogens for nutrients and space. Okay, we have a lot of organisms that we consider part of our normal microbiota that live on our skin. It's very difficult for other more pathogenic organisms to gain a foothold because they would have to outcompete all those organisms that are already there. We don't really understand why it is that our immune system allows some bacteria and viruses and protozoa to be there and not others. But we do know that our normal microbiota play a big role in stimulating. What I mean by second line of defense is, once it gets past the skin, for example, things like our immune cells and, other proteins and things that we have. Again, they've done really cool things with mice and raising mice in sterile environments. I don't even understand how they can do that, but once the mouse babies are born, again, they can keep them in a sterile environment. They sterilize their food and their bedding and all that stuff. And experiments that they've done with that helps us understand that these kind of mice can't fight off when they are exposed to pathogens. So we'll pick up there next week. Have a good day.